Brian's a second-year PhD student in our lab and is actually doing his advanced to candidacy exam not too long, in just a couple weeks. But he really has a background in bioengineering. So he was in our undergraduate program, and we were excited to convince him to stay for his graduate studies. So he worked with Andrea Casco, looking at polymer systems, and has turned that into thinking more about computational analysis of therapeutically relevant things. And that's really what he's going to show today. So some models, building models of cell signaling to figure out how we can develop a better drug.
All right, is that my cue?
Yeah.
All right, well, thank you very much for the introduction, Dr. Meyer, and thanks for organizing this, Ms. Barron. So like Dr. Meyer said, I'm a second-year PhD student in his lab, and the title of my talk and what I'm going to be telling you guys today is about how we used a simple two-step equilibrium multivalent binding model to predict cell type specific responses to IL-2 and IL-2 muteins. So fantastic. Okay, there we go. So I wasn't expecting such a nice introduction from Dr. Meyer, but I do have a background slide. So just to briefly go through that, I'm from Madison, Wisconsin. Like Dr. Meyer said, I did do my undergraduate education here at UCLA. I'm proud to still be a part of the bioengineering department. And he already covered some of my work with Professor Andrea Casco, but I also did some work with Professor Daniel Ennis in the magnetic resonance research labs. And he's currently at Stanford. And also the seminar series requested that we give you guys a little bit of background or a little thought about why we got into computational biology, which I'm happy to do. So that was actually my strongest introduction to this field was through a course taught by my advisor, Dr. Aaron Meyer. So that class focuses on the use of data-driven modeling to elucidate, visualize, and ultimately utilize the latent structures in the biological data sets of ever-growing size that we're all getting used to working with. So that's how I got interested in it. And if anybody in the audience is still taking classes, that's BE 175, 275. I highly recommend it. But enough about me and on to some of the background for the research. So like I said, we are going to be focusing on IL-2 today. And IL-2 is a member of the common gamma chain family of cytokines. This family of 6 cytokines, IL-2, 4, 7, 9, 15, and 21. And all these cytokines play really powerful roles in the regulation of both the adaptive as well as the innate immune systems. So why they're actually referred to as a family is pretty well demonstrated by this graphic. So each of these cytokines will first bind to a private receptor on an immune cell surface, but before signal transduction will actually take place, that receptor ligand complex will sequester gamma chain. Gamma chain is shared between each of these signaling pathways, and once this complexation occurs, the intracellular JAK domains on the private receptor and the gamma chain receptor will come together, and signaling will transpire through the JAK-STAT pathway. So like I said, these are really powerful and really important regulators of the immune response. And to kind of drive that home, I've compiled some phenotypes associated with loss of function in the genes encoding some of these either receptor subunits or signaling species. And in particular, we see that loss of function in the gene encoding the gamma chain, which again is shared between all these cytokines, results in a phenotype resembling X-linked severe combined immunodeficiency disorder. And you see a relative lack of T cells, B cells, as well as NK cells. So again, just to drive home the importance of these signaling pathways. So due to this powerful regulatory ability, IL-2, as well as the rest of the gamma chain cytokines, have attracted really broad interest as potential immunotherapies. In fact, IL-2, when it was discovered in 1976, was actually the first cytokine to be used in an immunotherapeutic setting. It's considered the father of immunotherapies. And we've seen considerable attempts to translate these regulatory abilities into a clinical setting. And when it was first used, it was primarily used as a potential anti-cancer immunotherapeutic. So there was significant attention when it was first discovered in 1976, and we've also seen a considerable resurgence the last 10 or 20 years into research and translating IL-2 into the clinical setting for reasons that I'll talk about in a bit. But the actual mechanism of action, which people were attempting to take advantage of when using IL-2 to treat cancer, is shown here. So high doses of IL-2 are known to proliferate CD8 positive cytotoxic T cells, as well as NK cells. And proliferation of these two immune cell types can lead to anti-cancer action through killing of tumor cells by the immune system. However, currently, despite all of the clinical trials, which I showed on the last slide, there are only two clinically approved IL-2 based therapies, and both of these are approved for the treatment of metastatic melanoma. So the reason for the really low success rate of these clinical trials is twofold. The first is that IL-2 causes significant toxicities in patients, can cause cytokine storm-like symptoms, as well as the sometimes fatal vascular leakage syndrome. And also the potency of this therapy is reduced by the fact that IL-2 will non-specifically activate immune cell types, and specifically in this application, it'll activate T regulatory cells. So T regulatory cells, we can kind of think of as the off switch for the immune system. Proliferation of this population will lead to the inhibition of effector cells through a variety of mechanisms, whether it be inhibitory cytokines or actually direct lysis and killing of effector cells. And of course, in an anti-cancer therapy application, this is a negative. You want to be having these tumor immune cell interactions. You want these effector cells to be attacking tumor cells. But what people have noticed in the past couple decades is that this selective activation of T regulatory cells can actually be harnessed and exploited to develop potential treatments for autoimmune diseases. And what people saw was that T regulatory cells specifically respond to IL-2 at low and intermediate doses of IL-2. So this specific or more specific proliferation of T regulatory cells leads to a net immunosuppressive effect, which can be used to treat a variety of autoimmune diseases. It's been explored to treat graft versus host disease, lupus, hep C-induced vasculitis. And this effect has been responsible for some of the resurgence in clinical translation of IL-2 in the past couple decades. So both of these applications of IL-2 face the challenge of attempting to proliferate some immune cells with the desired mechanism of action and avoid proliferation of off-target cells. But the challenge which is introduced here is that all of these cell types have the machinery necessary to respond to IL-2. So we quantified the amount of IL-2R subunits on each of our, or on a variety of cell types of interest. And what we see is that they all express significant amounts of the receptors necessary to transduce signals through the IL-2 pathway. So this data is kind of a lot to look at. I think some of the important trends that we see here are best summarized through a PCA plot. So on our scores plot here, this is just PCA of that same data, which I just showed you. On our scores plot, we have our different immune cell populations. And on our loadings plot, we have our gamma chain receptor subunits. And the important trend, which we're going to be looking at, is that T regulatory cells, which are shown in this cluster, through their high correlation with PC2, are shown to express relatively high amounts of IL-2R alpha. And IL-2R alpha is a necessary subunit for forming the trimeric high affinity receptor for IL-2. And then NK cells, which we can kind of think of, at least in their action, as the opposite of T regulatory cells, through their high correlation with PC1, are shown to express high amounts of IL-2R beta. So this general trend has driven quite a significant amount of the therapeutic development and protein engineering of IL-2. and has led to kind of this generalized trend, which I'm showing here, which is for the application of developing anti-cancer therapeutics and immunostimulatory IL-2 based therapeutics. People have attempted to develop IL-2 ligands with enhanced affinity for IL-2R beta and reduced affinity for IL-2R alpha. So this will selectively expand and induce signaling in CD8 positive populations, NKs, T helpers, all of our effector cells, while theoretically avoiding signaling and proliferation of T regulatory cells. And then, of course, the opposite is also true. So people have attempted to develop better autoimmune therapies through the development of IL-2 ligands with enhanced interactions with IL-2R alpha and reduced or ablated interactions with IL-2R beta. And the way that people have done this has been varied. There's been some really cool protein engineering technologies employed here. So there's been the development of IL-2 superkines through point mutations and structure-based engineering of IL-2 to enhance or reduce binding of IL-2 to the different receptor subunits. There's been fusions of IL-2 to receptors, pegylation of IL-2 to increase its half-life because IL-2 is generally cleared very rapidly in the blood. And there's also been the application of antibody IL-2 complexes. And most of these technologies so far have been based or are generally have their end goal of either reducing or increasing affinity for IL-2R alpha or IL-2R beta, depending on the desired therapeutic effect. So we're going to be kind of zooming in on one of the technologies which has been employed here, and that is the conjugation of an IL-2 to an FC fragment of a human antibody. So this was originally done in order to extend the half-life and improve the pharmacokinetics of IL-2, but naturally led to the conjugation of multiple IL-2 ligands to a single FC fragment, much like a normal human antibody has two antigen binding sites. So this creates what we are going to talk about, or well, what is a bivalent or multivalent IL-2 ligand. And in order to explore the effect which a lot of these affinity mutations and multivalency has on actual cell type specific signaling, we gathered PBMC immune cell responses to a really large panel of IL-2 muteins. So what we have is a comprehensive response data set measuring pSTAT5 response, which is the principal downstream product of IL-2 signaling. And we collected PBMC response at 4 different time points, 12 different concentrations to these 13 mono and bivalent ligands. And we're also happy to work with a panel of ligands with altered affinities through these point mutations, representing a wide variety of affinities, both for IL-2R alpha and IL-2R beta subunits. So because we have this highly standardized data set at 4 different time points, 12 concentrations, 4 cell types, and a variety of ligands. The first thing we did was attempt to use a structured decomposition method to actually break down and visualize some of the signaling trends here. So we formed our data into a tensor. And in this case, it's a 4-dimensional tensor, which is summarized here. Again, the dimensions of this tensor are time, ligand, cell type, and concentration of cytokine. And then we used CP tensor factorization to break down this tensor into components. So this is a dimensionality reduction technique, tensor factorization. You can kind of think about it as n-dimensional PCA, and it allows us to map the correlation which each of these dynamic effects have with specified components and visualize some of the trends in the signaling data that we have. So we saw that with this really large set of data, using only 3 components, we're able to explain over 90% of the variance contained in the signaling data set. And then we're able to map the correlation of our ligands as well as our cell types with each of these components. So of course, the results here are a lot to look at. So the thing that drew our attention and that I'm going to draw you guys' attention to is that our bivalent ligands even above their monovalent counterparts, have a high correlation with this second component. And what that means is we can go over and look at our cell types and their correlations with each of these components. And we can see that T regulatory cells also have a high correlation with this component too. So what this is telling us is that our bivalent ligands are selective for T regulatory cells, which again is a really key performance metric for the development of potential autoimmune treatments. Since concentration was another dimension in our four-dimensional tensor, we can also look at which points in our dose response curves this specificity is dominating. And what we see is component 2 explains the most variance in the data at low and intermediate concentrations. So to summarize the exciting results which we're getting here, we're seeing that bivalent IL-2 muteins are highly specific for T regulatory cells, and that the specificity is most pronounced at intermediate and low concentrations of cytokines. So, of course, having seen that and having uncovered this trend using an unbiased tensor factorization method, the next thing we asked was, well, why is this happening? Why are these bivalent ligands, even above their monovalent counterparts, both conjugated to an FC fragment, enacting this selective response in T regulatory cells? So to take you through some of our thought process and to kind of play devil's advocate for the rest of the findings in this talk, I'm going to go through some of the possible effects which we identified. And the first is, of course, the difference which multivalent ligands have in surface receptor kinetics over their monovalent counterparts. So multivalent ligands differ from monovalent ligands, of course, in that they can engage multiple receptors simultaneously. And so they will ultimately bind to a cell surface with an altered affinity in comparison with their monovalent counterparts. And this functional affinity is known as avidity and has been shown to change with the actual, or be a change as a function of the abundance of receptors on a cell surface. So the affinity is dependent on the actual expression levels of these receptors, which is not true of monovalent ligands. We also wondered whether multivalent ligands could be transducing signal in an altered fashion. So perhaps the clustering of active receptors or active complexes would lead to clustering of intracellular components, perhaps stronger signal transduction. This is known to happen in B cell receptors, T cell receptors, or in FC gamma receptors. And we also wondered whether potentially multivalent ligands were being trafficked in a manner different than monovalent ligands were. So potentially, or it's been shown that multivalent ligands are endocytosed using different cellular machinery in relation to monovalent ligands. But so these are a few of the effects which we identified and wanted to think about, but ultimately we decided that we wanted to model and explore the effects which altered receptor ligand interactions may have on downstream effects, and see whether we could explain the differences between monovalent and bivalent signaling purely through surface receptor ligand interactions. And the way which we wanted to do this was through use of a equilibrium multivalent binding model, which is summarized here. And this binding model functions by, of course, any monovalent ligand will interact with a receptor on a cell surface with a certain association constant. But this model allows us to predict the binding of heterovalent ligands and multivalent ligands. And it does this by making the key assumption, which is summarized here. The model assumes that the initial interaction of a multivalent ligand complex proceeds with the same association constant, which the monovalent monomer will interact with the receptor. But that subsequent interactions, which of course is the key characteristic which differentiates multivalent binding from monovalent binding, is that secondary, tertiary multivalent interactions occur with the same association constant, but modulated by a kx star parameter. So this kx star parameter encapsulates a whole lot of physical phenomena. But in this particular context, you can kind of think about it as the propensity which these multivalent ligands bind multiple receptor subunits. So to summarize our actual modeling approach here, the inputs to our model is simply our ligand valency format, as well as the receptor affinities for these ligands, which we've already quantified for each of our IL-2 muteins. The model also takes in the abundances of receptor subunits on each of these immune cell types, which we've also quantified. The calculation which is actually occurring here is the steady state or equilibrium amount of active surface receptor complexes. So this is known biology. The active signaling complex in the IL-2 pathway is constituted by a gamma chain, an IL-2R beta bound by an IL-2 molecule. So we calculate that. And then the only fitting which is actually occurring here is we optimize a kx star parameter for our bivalent ligands. And we also have to use a scalar parameter or a scalar factor, which we're fitting to convert from the amount of equilibrium active signaling complexes to p-stat MFI. So it's a very minimal fitting procedure. So just to back out and give a little bit of a bird's eye view of this approach to modeling signaling in response to bivalent ligands, I just wanted to show this kind of schematic of how we're thinking about it. So naturally, the steps which are necessary to actually transduce a signal and ultimately leads to proliferation of immune cell types in response to IL-2. You first have necessary ligand receptor interaction in the IL-2 and Gammachain pathway. You then need dimerization of receptor subunits. That leads to intracellular signaling events, the phosphorylation, transnucleation, dimerization of STAT proteins. And finally, that actually leads to a change in gene expression and completed signal transduction proliferation. So our modeling approach, which is simply calculating equilibrium binding on the cell surface, is kind of cutting out the middleman. It's a much simplified approach to looking at how multivalent ligands actually induce a response in immune cell types. But the simplified approach actually results in a model which is able to very accurately reproduce cell type specific responses to IL-2 muteins. So the results of that fitting process are summarized here. This is all of our experimental p-stat5 results against our model's predictions for the p-stat5 levels in response to these cytokines. This is at all of our different concentrations at the 30-minute and hour time point and for our panel of cell types. And some of the key assumptions, again, are shown here. According to our understanding of the pathway biology, each of our IL-2 ligands in our binding model is allowed to bind to one IL-2R alpha and one IL-2R beta receptor. Again, this only fitting which is happening is conversion from active signaling complex to p-stat5. And this is done on an experimental run basis. So this R-squared value of 0.89 is really exciting. It's a very computationally inexpensive model. And we also see that this accuracy is preserved across cell types. So it's able to predict T regulatory cell, T helper, and CD8 cell pretty accurately. And the accuracy is also preserved across our panel of ligands, both for the monovalent and bivalent formats. So I think that the true utility of computational modeling in the context of modeling signaling pathways is ultimately we want to be able to guide the design of even more effective ligands, not just understand what's going on with our current signaling data. So what we next did was use our model to optimize the selectivity of ligands for particular cell types and explore whether the selectivity conferred by bivalent ligands over their monovalent counterparts could be pushed even further with ligands of higher valency. So the optimized or the activity of these theoretical ligands was optimized for T regulatory cells in this case at each different valency. And the activity of each of our cell types of interest are shown normalized to their optimal activity at a valency of 1. And what we see is that our model shows that selectivity for T regulatory cells can actually be pushed a lot further, theoretically at higher valency formats. Our model also shows that in order to actually achieve this optimal selectivity for T regulatory cells, coordinate affinity changes have to occur. So the affinities, which must be conferred to IL-2 to achieve these optimal selectivities at each of these valencies is shown to our right. And in agreement with generalized protein engineering trends, our model shows that for higher selectivity for T regulatory cells, you want to reduce IL-2R beta affinity, and actually shows that you want some intermediate value of IL-2R alpha affinity in order to get this selectivity. We also then went ahead and saw whether this selectivity for NK cells could also be predicted and potentially pushed further at higher valencies. And we saw, or we recapitulated the results that potentially higher valencies could confer even greater selectivity. Again, here, coordinate affinity changes are needed. Some intermediate value of IL-2R beta affinity must be achieved to get the selectivity at higher valencies, and ablation of IL-2R alpha interactions is also necessary. Finally, we also have IL-7R alpha quantitation. So we plugged it into our model to see whether we could selectively activate T helper cells using IL-7 muteins of higher valency. And again, we saw higher valency could confer greater selectivity. So in conclusion, the story here is that we first saw that bivalent IL-2 muteins selectively target Tregs, and we visualized this using tensor factorization methods of a really comprehensive signaling data set. We then saw that a simple multivalent equilibrium binding model was very effectively able to translate between binding and actual downstream pStat5 signaling for all of these muteins. And we used this model to explore the potential benefits which ligands of higher valency may confer in enacting more selective signaling and ultimately more effective therapeutics with reduced off-target toxicities. So thank you to everyone for listening, and to everyone who's worked on this. Of course, my advisor, Dr. Aaron Meyer, as well as some of the graduate students who worked on collecting the data. Special thanks to Cyrillus Tan. He's done a lot of great work in building this and improving this equilibrium binding model. So if you guys have any questions, I'd love to take them at this time.
That was awesome. That's pretty amazing and impressive.
Thank you.
So the relationship between, it seems to me that the relationship between binding and downstream STAT5 is fairly simple, right?
Yes. So at shorter time points, it's generally thought of as a linear relationship. But potentially at longer time points and more complex dynamic activation, there is STAT phosphorylation, dimerization, transnucleation, and there's some more important downregulation of signaling at longer time points, which our model is admittedly not including.
But in the short term, okay, so that's a good start. And then I guess, so for the scope that the model is in now, where do you think are the remaining challenges or deficiencies?
Well, that's a great question. So in this context, we are really excited about what it can currently do.
Yeah, it looks perfect. That's why I'm asking the question.
Yeah, no, absolutely. So I think that I mean, the deficiencies are, of course, in its simplified approach. So I think in this context, I don't know how much further it can be pushed, because if we want to include longer time points, it's not a dynamic model, and we can't include those effects or those dynamics. But the advantages, of course, of these simplified look at how signaling is actually occurring, we think is useful for initial stimulation of these immune cell types. So in its current format, I don't know if there's a whole lot more that can be done. I do think that we can apply this very inexpensive approach to explore potential benefits of multivalency in other pathways which are characterized by pleiotropy. But as far as how much further we can push this exact model in this context, I don't have a clear idea of how that would be done, because it would require significant modeling approach changes.
Mm-hmm. No, yeah, that sounds great. I mean, I think looking at connecting it to the actual biological effect is going to have it's a whole other set of questions, right? So, and longer term, it's dynamic, and it's a cell type specific regulatory network that controls that. So,
yeah.
So, great. Any other questions?